You'll find that, like our skeletal tissue discussion, muscle tissue also deviates from our lab material quite a bit. Just like we did with skeletal, we'll be focusing primarily on microscopic muscle anatomy and the physiology of muscle function, while we leave any muscle naming to lab material. When we look at what we're meant to cover, we do have to get into cardiac and skeletal muscle, at least briefly, because those are two of the three types of muscle tissue. But we are quickly going to be getting into a skeletal muscle focus. We're going to be looking at the types of fibers, the composition of the skeletal muscle cell, and the arrangement of the interior of the muscle cell that allows for that cell to shorten. Then finally, we'll get into some fiber organization, the effects of exercise on skeletal muscle tissue, as well as the mechanics, the biomechanics of muscles and how they orient around joints. The main reason we're focusing on skeletal muscle tissue is because you will see cardiac at quite a bit of in-depth manner when you do cardio. There you'll talk about the heart, which is primarily a cardiac muscle tissue organ. So you'll have plenty of time to get into that muscle tissue type. Smooth muscle, we'll be talking about smooth muscle kind of here and there throughout A&P 1 and 2. But again, you'll talk about smooth muscle most in depth when you get into both respiratory and cardiovascular. So as mentioned, three types of muscle tissue: skeletal, cardiac, and smooth. And the easiest way to look at them is to basically look at similarities versus differences. First and foremost, we can take these three and use two variables in which to describe them. Their gross physical appearance on a microscope slide and our voluntary nature of control. On a slide, each of these types of cells will either look striped or unstriped. And the term we'll use to describe that is striated. Striated has a striped appearance. So it'll be striated versus non-striated. Of the three types of muscle tissue we have, both skeletal and cardiac are of the striated variety. So when you look at them under a microscope, they will appear striped. Smooth is given its name because of its lack of striations. It has a smooth appearance to it. The second variable will be its voluntary control. Skeletal muscle is the only one that is going to be considered voluntary muscle tissue. The other two will act as part of either an automatic process in which we have no voluntary control over, or it'll be part of autonomic reflexes. So we'll look at cardiac and smooth briefly, so we can use them to compare and contrast with skeletal, but we will not be getting into cardiac and smooth in any in-depth manner. Cardiac muscle cells are striated and involuntary, as we said earlier, but they're also short and branched. You will see that as a main characteristic difference between it and skeletal. Skeletal muscle fibers are long, parallel bands that run from one end of the muscle organ to the other. Cardiac are short. They're attached end to end, and they have the ability to form a branching network. Each of the attachments that we see in cardiac muscle is called an intercalated disc. That's going to be the joint, essentially, between one cardiac muscle cell and its neighbor. Buried within those intercalated discs are another type of cellular junction called a gap junction. These are protein tubes embedded in that intercalated disc that's going to allow for easy exchange of material from one cardiac muscle cell and its neighbor. Because of those gap junctions, we are able to get an autorhythmicity. And it doesn't mean we have to have what we call an independent irritability. In a skeletal muscle that we'll talk about later, every single muscle cell has to have a neuron telling it what to do. Not so for cardiac. Because of those gap junctions, if we contract this cardiac muscle, then subsequently its neighbor will contract. And then its neighbor will contract. So as long as we stimulate one particular area of that cardiac muscle organ, we will see a wave of contraction spread through that mass of tissue. That combined with our autorhythmicity and the branching makes it perfect for the heart. We have a sac of blood that we want to contract and eject as high a volume as possible. The branching network that we see with these cells allows for a three-dimensional contraction of the heart as a whole. The intercalated discs with the gap junctions allow us to only have to innervate one part of the heart and get a sequential contraction that goes throughout the heart. And then its autorhythmicity allows us to have a base heart rate within that organ that we can count on and then speed up or slow down as needed. Smooth muscle, again, lacking striations, involuntary as far as its action goes, looks like a completely different structure altogether, although it does seem to have some similarities once we look a little closer. First of all, yes, they're shorter, but they take on this kind of oval-looking shape. And when they contract, they don't just shorten, they kind of contract in upon itself and change three-dimensionally. This is because the nature of the arrangement of the fibers inside that muscle cell. For skeletal and cardiac, they were running parallel, and that's what gave us the striped appearance. For smooth, we tend to see the overlapping fibers run at angles, using dense bodies as anchor points. And it allows each individual cell to get a three-dimensional contraction. We tend to see smooth muscle cells arrange itself in a circular manner around things like ducts. So when we contract those cells, the duct will open and close, expand and contract, dilate or constrict based on the actions of those muscles. So now let's turn our attention to skeletal. Striated voluntary muscle tissue. This is the muscle tissue that we think of when we think of our musculoskeletal system that involves locomotion. Now, every type of muscle tissue will have these four properties, not just skeletal, but from here on, again, we're focusing on the skeletal. There has to be an excitability. There has to be an ability for that muscle cell to respond to a stimulus. We said that skeletal muscle cell actually has an independent excitability. Every single muscle cell has to have a neuron stimulating it to get it to contract. Once it's excited, it has to have a contractility. So its excitability leads to its contractility, the ability to shorten. And then lastly, we kind of look at the last two together. It has an elasticity and an extensibility. I say this when we bring up elasticity in a couple different systems or topics. Elasticity does not mean the ability to stretch. Elasticity means that if we do stretch something, and then we remove the forces that stretched it, elasticity means it springs back to its original length. And muscle has elasticity. If you stretch a muscle, the connective tissues running through that organ apply a counter force. As soon as you relax the muscle and remove the stretching forces, it will respond and spring back to its resting length. But conversely to that, it also has to have an extensibility. Every muscle has what we refer to as a resting length. We have to have the ability to stretch beyond that resting length. So in a way, we kind of look at these four properties and we can kind of group them together. We have an excitability and a contractility, because if we can excite the muscle, then we will express its contractility, and then we have to have an ability to extend the muscle, but then as a part of that, we get a reflex of elasticity. Each muscle, skeletal muscle, is considered an organ in and of itself. It's going to contain all four of our tissue types. In addition to the muscle, we have neural tissue, not only providing sensory or motor input to get it to contract, but also getting sensory information back about the position, length, stress, and strain on that muscle. It is a striated tissue, and it usually attaches to bone. We are going to consider this to be a skeletal muscle tissue. However, by the time you get to this lecture, you likely will have already had a lab involving the facial muscles, several of which have no bony attachments whatsoever. Some attach to bone on one end, but then soft tissue on the other. So think about the muscles that kind of blink the eye, the muscles that purse the lips into a puckering movement. There is no bony attachment for those particular muscles, but they still fall under our skeletal muscle structure and the way it contracts with its excitability voluntarily. The only difference is it's not directly involved in locomotion. Skeletal muscle does take on a number of functions, right? Not just locomotion, as we saw with the facial muscles. Body movement is going to be the calling card, and hand in hand with that goes the maintenance of posture. We rely on skeletal muscle tissue for temperature regulation. Heat is a typical byproduct of muscle activity. It's metabolic activity. And as a result, we've all experienced times when body temperature has started to drop, and we shiver as a result, a quick fluttering of the skeletal muscle as a way of increasing its metabolism and generating heat. We see muscle used as a storage for certain things like myoglobin, small storages of glucose for muscle contraction in the short term, be stored in the muscle. We also use it for movement. When you do cardio, you'll study what's called the skeletal muscle pump. Veins that go through the skeletal muscle that now when you contract the muscle will pinch the blood vessel and move blood through the circulatory system in times of low blood pressure. Now, when we look at the arrangement of skeletal muscle, it's easy to get lost in the diagram. So make sure you know the terms first, then apply it to the diagram. Sometimes I'll recommend you do the opposite. Study the diagram first and learn the terms that way. But I recommend you learn the terms first, the definitions, and then you apply them to the image. Each muscle is comprised of bundles of muscle cells called fascicles. And what we're doing here now is we're taking a step down and getting more and more and more microscopic with our tube-like structures. So picture this long muscle, and within it are long, parallel, tube-like fascicles. In each fascicle is a number of long, parallel, tubular muscle fibers. In each fiber are long tubular structures called myofibrils, and then each myofibril is made up of long proteins called myofilaments. So we're stepping down, looking very similarly as we get more and more and more microscopic. So now let's apply that to the diagram. Here is our long muscle organ coming off the bone. We do a cross section of it. This is a fascicle. Each of these subunits within the muscle is a fascicle. That's a bundle of muscle cells. Now, if you hear me use the term muscle fiber, that means the same as muscle cell. Those are perfectly interchangeable. So here now we have an entire fascicle being extended out of the muscle. A fascicle, again, by definition, is a grouping of muscle cells. So this that's extending out of the fascicle is a skeletal muscle cell. Extending out of it is the myofibrils. The myofibril is a long tubular structure that runs the length of the muscle cell. The myofibril is going to be the smallest growth structure that will undergo the ability to shorten. When we talk about myofilaments that make up the myofibril, those are shorter, and the myofilaments do not shorten themselves. How we get overall gross muscle shortening is by overlapping myofilaments and witnessing them slide over one another, giving us elongating and contracting myofibrils. And if we shorten the myofibrils, we'll be able to shorten the muscle cells. And if we shorten the muscle cells, we'll be able to shorten the muscle organ. Interspersed through that structure, we had three concentric connective tissue layers. We had our endomysium, endo meaning within. It's the innermost. We will see endomysium in between the muscle cells, giving us electrical insulation. Remember, this is an excitable tissue. So if we were going to excite this particular muscle cell, we don't want that electrical input to affect any of the neighboring muscle cells. We want to have specificity over what we contract. So separating each muscle cell within a fascicle is going to be a layer of this endomysium. Surrounding each fascicle is perimysium. So you'll see this perimysium in between each of the fascicles, subdividing the muscle up into those fascicles. That's going to be there for protective purposes. It helps support the muscle as a whole, and it's going to be part of that connective tissue structure that gives muscle its elasticity. Surrounding the superficial aspect of the muscle, and you can see here on this diagram that it's kind of peeled off the image. That's epimysium. Epi meaning outer or around. We get a connective tissue layer surrounding that entire muscle organ. So if you were doing a dissection and you cut through the skin and the adipose tissue of the subcutaneous layer and you got down to the muscle, this is what you would see on the outside of that muscle. Now, each of those three connective tissue layers is continuous with one another. And they're going to emerge from the end of the muscle as the tendon. So picture, we've got our endomysium extending in between the cells, the length of the fascicle. We got the perimysium surrounding the fascicles, but it's running the length of the muscle. Well, by the time we get to the end of the muscle here, all three of those layers converge together and form this dense connective tissue band that will attach it onto the skeletal system. Now, outer from that muscle, exterior to the organ, we do get some other connective tissue layers. And while they're not part of that muscle organ, they do play a role. We get deep fascia. Deep fascia will separate one muscle from another muscle within the same group. So picture your quadricep muscles and your anterior thigh. 4 muscles there as a group. You will find deep fascia in between each of those muscles. Superficially, we will find the superficial fascia, separating the muscle from the skin. Now, there was a time when we thought that this fascia was nothing more than packaging. However, the more we look at the arrangement of the fascia and the structure of the fascia and how it responds to activity, we consider the fascia now to be a great, effective, reflexive, communicative system throughout that muscular system. we see the fascia constrict and relax, not in the same way we see muscle tissue constrict and relax, but realize that if we want our muscular system to perform efficiently as a whole, we're going to rely on a lot of synchrony of movements. We no longer want to picture muscle movement in the sense of very simplistic movement, even though we'll use some of those simple movements to describe and illustrate muscle function here, like picking up a dumbbell and curling it. Extremely simple movement using one joint. But picture the muscle synchrony that has to be in place to pick up a golf club and swing and hit the golf ball perfectly. Every muscle has to be in perfect sequence of contraction for that degree of coordination. Think about picking up a baseball and throwing it, and how every muscle has to fall into that sequence perfectly for an accurate throw. So we want to have some sort of reflective communication system throughout this muscular system, and we rely on the fascia for that. When you jump from a height onto the ground, yes, it's going to be your calves that are going to start bearing that weight first, then your quads, then your lower back. We want the quads to be ready as the calf muscles are contracting. We want the low back muscles to be ready as the quads are contracting. So as soon as your feet hit the ground, you see a wave of vibration spread through this fascia, preparing the next set of muscles in sequence. So it helps us effectively time synchronous muscle movements. When we get more and more microscopic, 'cause that's kind of how we arrange this lecture. We start off with a gross muscle look, and then we get smaller and smaller and smaller. When we look at the muscle cell, we have to introduce some new terms. We started off early in this class, not this lecture, but this class as a whole, talking about the structure of the cell. We talked about the membrane, the nucleus. We talked about ribosomes, and endoplasmic reticulum, and Golgi apparatus, and so on. We talked about a generic cell, which really, that wasn't really a complex enough discussion to talk about any specific cell, right? Even by now, you've talked about a lot of very specific cells in lab. Osteocytes and chondroblasts and so on. Red blood cells. When we're talking about a specific cell, very often we have to look at what that cell does functionally, and certain structures within that cell may take on a completely different function than what we described in our generic cell. And if it does, we have to give it a modified name. That's what we see in skeletal muscle. We see two structures in skeletal muscle that weren't in our generic cell at all, and we'll get to those. We've got to add those. But we also see three structures that we did have in our generic cell. But in our generic cell, they didn't take these functions on, so we have to give them an updated name. First off, in our generic cell, we had the plasma membrane, and we spent more time talking about the membrane than we did any other part of that cell. Well, in the muscle, the plasma membrane takes on a very specific role. We are going to talk about how that plasma membrane conducts an electrical stimuli across its surface, which leads to muscle contraction. So therefore, new name, sarcolemma. When you hear me use the term sarcolemma now, I'm referring to the plasma membrane of a muscle cell. In a generic cell, we had cytoplasm, the internal components of a cell. We had cytosol being the liquid. We had cytoplasm being the entire internal structure. Well, here, slightly new function, sarcoplasma. we need to have a high calcium component in our cytoplasm of a muscle cell in order to undergo contraction. So therefore, we call it sarcoplasm. And if you're wondering where that calcium comes from, it comes from a modified, smooth endoplasmic reticulum, which now we're going to change the name to sarcoplasmic reticulum because of the calcium. So there's our three modified components: sarcolemma, sarcoplasm, and sarcoplasmic reticulum. The new structures added are transverse tubules. This is an extension of our sarcolemma, our membrane. So picture you're extending across the surface of the cell and then you see a hole. In this case, that hole is a tube leading from the surface of the cell down deep into the cell. The second addition is called a terminal cisternae. They are going to be blind sacs that are part of our sarcoplasmic reticulum. And they will run parallel with the transverse tubules that extends down from that cell membrane. So together we get a transverse tubule and we get two terminal cisternae. We're going to call that structure the triad. Now picture these two structures as a functional bridge. I'm going to put the cart before the horse just a little bit because we won't get to talk about muscle contraction until the next lecture. But in order for us to have muscle contraction, we need to have an electrical stimulus on the muscle cell membrane, the sarcolemma. Down deep in the muscle, we have to have calcium released from our sarcoplasmic reticulum. We have to make sure that the electrical stimulus on the membrane somehow innervates that sarcoplasmic reticulum to release its calcium. We do that by connecting both pieces of that anatomy with our triad. We got the T-tubule, that's continuous with the membrane. That allows that electrical stimulus to get deep into the cell. We got the terminal cisternae, which is an extension of our sarcoplasmic reticulum, which contains the calcium. So functionally, we get our impulse go along the sarcolemma, down the transverse tubule, and it causes the terminal cisternae to release calcium from the sarcoplasmic reticulum, kind of connecting those pieces of anatomy. Now we can move on with the rest of our contraction lecture. Within the cell, we have our myofibrils. Remember, those are the long, continuous, cylindrical structures. Myofibrils shorten. And when the myofibrils inside a cell shortens, the cell as a whole will shorten, then the muscle as a whole will shorten. But inside the myofibrils is where we're going to spend more of our time talking, the myofilaments. These do not run the length of the myofibril. They're shorter. But these are the ones I mentioned earlier that overlaps with each other. And as they slide back and forth, that's the mechanism that gives us our overall shortening. So these myofilaments will stay the same length the whole time. The structure they make up will change its dimensions. We've got two general types, a thick filament and a thin filament. The thick filament we're going to refer to more so as myosin. Myosin is the thicker of the two, and it's actually going to have an angled projection. And we're going to refer to it as the myosin head. We use the myosin head to form cross bridges, attachments with our thin filament. The thin filament we're going to refer to as actin, A-C-T-I-N. So in order for us to get contraction, we need the myosin to form those cross bridges with the actin. But at rest, there are two proteins that take on a regulatory role and prevent the myosin from attaching to the actin. This is troponin and tropomyosin. Tropomyosin runs along with the thin actin filament, physically blocking access of the myosin head. As long as the tropomyosin is sitting in the way, myosin can't bind to actin. Troponin sits on the tropomyosin. Troponin binds to calcium. Now, we mentioned calcium earlier. We said we needed calcium to be present for us to have contraction. Well, when we release calcium from our sarcoplasmic reticulum, it binds to troponin. The troponin then changes shape and pulls the tropomyosin out of the way, exposing the binding sites on the actin. Once we look at our contractile lecture, we'll look at this in just a little bit more detail. But the fact that these sit in between the actin and the myosin, preventing them from attaching, that's what gives these two proteins a regulatory role. Because as long as they sit there, that muscle will not shorten. So here's a pictorial representation of what we have. Here is a bundle of those myosin filaments. See those angled heads sticking off. This is the part that's eventually going to attach on to the actin. The red chain is your actin. The red chain is your actin. Now, sitting with it, you see the tropomyosin and the smaller, more spherical troponin that's attached to the tropomyosin. Again, calcium will bind to the troponin. Troponin will change shape and pull tropomyosin away. Now we can form these cross bridges between our actin and our myosin, and we can get muscle shortening as those cross bridges do their thing. Now, when we look at those two filaments, this is the arrangement that gave striated muscle its striped appearance. You can see how we have areas here where we have more dense overlap and other areas here where not quite as dense overlap. If we shine light through that, we're going to get these alternating light and dark bands. That's what gives us our striped appearance. But this structure is also what gives us our ability to shorten. The sarcomere is going to be the smallest contractile unit of a skeletal muscle. And the easiest way for us to look at this is by simply jumping to a diagram. And we will start highlighting the individual pieces as we go. Here, to orient ourselves again, here's our myosin filament. Here's our actin filament. The sarcomere is the span from one Z disc to another Z disc. That is the boundary for our sarcomere. The Z disc runs perpendicular to the sarcomere, and we get attachment with the actin filament on either end. Because of the arrangement of the actin and the myosin, the light and the dark bands, we can divide this sarcomere up into bands and zones itself. Now, when you're studying these, it's important to be able to identify them on a drawing like we have here in front of you now. But it's also important to know what's in each band. Because as we shorten, As we take this Z disc and we start pulling it in that direction, and we take this Z disc and we start pulling it in that direction, you will see some of these bands and zones change their dimensions and others won't. So it's important to know what we find in each band. So let's look at the H zone. That's a great place to start. The H zone is the part of the sarcomere where all you get is myosin. It's from the tip of this actin to the tip of that actin. So all you see in the H zone is myosin filament. The A band is the next place you go. While the H zone only had myosin, it discounted the places where they overlapped. The A band goes from one end of the myosin to the other. In between consecutive A bands, because remember, these sarcomeres are going to be linked end to end to end like boxcars on a train. In between each consecutive A band, we have an I band. The I band will only contain the actin, and it will also contain the Z disc. So that's our major players. Now we just need to go through two more parts of this to kind of clean it up. First off is the M line. That runs right down the middle of our sarcomere, perpendicular to our myosin. Now, a lot of people will get the impression, and some of the definitions don't help, the M line will be described as the center of the sarcomere. And it is the center of the sarcomere. But they talk about it almost like they talk about the equator on a map. Obviously, if you've ever gone into the southern hemisphere, you didn't cross some big dotted line on the map. It's drawn on the map. It doesn't exist in reality. In this case, the M line exists in reality. It's a protein. It's a protein that runs perpendicular to our myosin filaments. And it helps anchor them into their orientation. It's the M line that makes sure these myosin filaments all stay in their place, running parallel. So it's not just a fictitious line indicating the middle of the sarcomere. It's a protein, happens to be in the middle of the sarcomere. The last is the protein titin. Now, I like this diagram in particular because they draw titin looking like a spring. That's kind of what it does. It prevents overstretch. Remember, where the actin and the myosin meet, we're going to see those two filaments slide over one another. Well, if we stretch this muscle, this titin protein will increase in tension. So now, when you remove the stretch from the muscle, it's this titin that's going to help bring that muscle back together a little bit. Because what we want to prevent is a loss of this overlap. We can only contract this muscle if the actin and the myosin are overlapping with each other. The minute we pull this sarcomere apart and there's no longer any overlap, we've completely lost the mechanism by which we can shorten that muscle. So we generate a tension in that muscle through those two filaments sliding over one another, which we call the sliding filament theory or the sliding filament hypothesis. We see the width of the A band remain constant, but if we shorten enough, the H zone will not only get narrower, but it completely disappear. The Z discs move closer together. So again, picture these sarcomeres attached end to end to end to end. Each one shortening just a little bit, giving us a significant shortening of the myofibril, which it's contained in. So now let's look at a couple slides and we can zip back and forth and look at how that sarcomere changes as we contract the muscle. So here we have an arm kind of at a bit of a resting length, slightly bent. Here's the orientation of the sarcomeres in that muscle in that position. And here is how it would look on a slide with the alternating light and dark bands, giving us that striated appearance. So you see here, Z disc, look at that light band there. That's implying that we're looking right through here where there's not a whole lot of density of structure. However, we move just a little bit to the left, it's darker. Now we got that myosin and actin overlapping, blocking a lot of the light coming through. So we stimulate that muscle. The sarcomere shortens, the actin and the myosin slides over one another, and we see our Z discs get closer together. So now let's go back and watch the sarcomere shorten again. We lengthen, we shorten. We lengthen, we shorten. See the H zone. As we lengthen, it gets bigger. As we get more and more filament overlap, the H zone gets smaller. Now we got a little bit more we can contract. We shorten a bit. Now we shorten even more. Now you can see how all that structure now is really starting to bind up on itself. We got nowhere left to go to get us more sliding. So for some muscles, even though it looks like the arm here now is limited not only by the muscle sarcomere, but also by the structure of the joint. But you can comprehend that there's a maximum amount that any muscle can actually shorten. And that's true. At this point, that muscle will not generate any continual force and get shorter and shorter and shorter. The H zone is gone. However, you notice the A band hasn't changed. The A band's always going to be consistent with the length of the myosin filament. And none of these filaments themselves change length. They just slide over one another to change the length of the sarcomere. Now, we said that each muscle cell has independent irritability. An ability for that muscle cell to constrict, we have to have a neuron telling it what to do. However, we rarely recruit just one muscle cell. We recruit them in groups called motor units. A motor unit is a single motor neuron plus all the muscle cells it controls. A single motor neuron plus all the muscle cells it controls. Now, a motor unit is going to be all or none. Either it constricts with as much force as it can gather, or it doesn't constrict at all. All or none. So let's look at how that applies now to generating force in a muscle organ. Here we've simplified it. And we've got two motor units, a blue one and a red one. We got a blue motor neuron coming from the spinal cord out to the muscle. The blue motor neuron is going to control these two blue muscle cells. We also have a red motor neuron, this time controlling these four red motor muscle cells. If we want to pick up something and generate a little bit of force, then we will use maximum contraction of our blue motor unit. Each motor unit, remember, is all or none. So we recruit the blue one, but they independently contract fully to give us a certain amount of force. We want to generate just a little bit more force. Now we relax the blue one, and we go full contraction on just the red ones. So we had two contracting before, now we've got four. And now if we want to generate just a little bit more force again, we use the blue ones and the red ones. So now in a muscle organ, obviously there's going to be a lot more motor units than this. If you want a muscle that undergoes a lot of strength but very little dexterity, you will have each motor unit comprised of a number of muscle cells. Whereas you might look at the muscles of the hand for very fine manual dexterity. Each motor unit might only have a couple muscle cells for fine control. When we talk about innervating the muscle, though, there's a couple of terms that I want to make sure that we get straight. Muscle tone and muscle contraction. We have to think of those as two separate events. And muscle tone has to be one of the misleading terms that we use here because it's used inappropriately in everyday language. Every muscle has a resting muscle tone. Even if a muscle is relaxed, there's a little bit of neurological innervation going through that muscle to keep it at a certain heightened ability so that if we want to get a contraction, it's at the ready and ready to go. This does not necessarily refer to people who say they have tense muscles. Their neck is tight. They got a muscle knot. That's not this. I will try and post some videos of Parkinson's. And you will see they have what's called a lead pipe rigidity, where they just cannot relax their muscles. And even if they're letting you move their arm and you're trying to move their arm, it'll feel like you're bending a lead pipe. There's going to be resistance in every single way. That's a heightened tone. When we innervate a muscle to get it to contract and shorten, that's the generation of strength, the generation of force. And we can take that and subdivide it even further. We can do an isometric contraction. Metric being a unit of length, right? Iso meaning same. We got a contraction that gives us no change in length. However, if the tension overcomes the resistance, then we can get an isotonic, where now the tension in the muscle is consistent, but the muscle shortens. If the muscle is shortening, that's going to be a concentric isotonic contraction. So you're curling a dumbbell up, that's concentric. Let that dumbbell back down. You still have to keep the muscle contracted as you lay it down. That's an eccentric contraction. And we see that on the diagram here. This lady picks up 80 pounds and it's a little too much for her. She tenses up her bicep, weight does nothing. We got a contraction here, but we did not get any muscle shortening. She goes back to the tens and she can dominate the tens. This time she gets a contraction here and the weight moves. That's our concentric isotonic. Now, as we watch muscle move, we have to give ourselves an orientation. We always designate one end of the muscle to be the origin and the other end to be the insertion. The origin end is going to be more fixed in place. The insertion end is the end that moves through space. So we look at our bicep curl analogy again. At the bicep, the shoulder would be the origin. And because the forearm is what moves through space, the muscle attachment at that piece of anatomy is considered the insertion. We can look at muscle fibers and categorize them in three different ways. Now, for us, we're going to be focused on the extremes, because very often any characteristic that we have on the extremes, this middle one literally splits the difference. Slow oxidative fibers. These are fibers that have a very high mitochondrial content. They are not very good at developing explosive, high-force movements. What they are good at are low-force movements that have to be sustained over a very long period of time. Picture the muscles of the core, the muscles that hold up the head. The neck muscles hold up the head weight all day long, yet they really don't feel fatigue. The muscles of the arms would be more fast glycolytic. You could pick up a baseball and hurl it 70, 80, 90 miles an hour. However, if you threw that ball 90 miles an hour and I handed you another ball and said, throw it again, throw it again, throw it again, they would fatigue quickly. If I asked you to hold that arm out to your side, no weight, just the arm, the muscles in the deltoid would start to burn and fatigue. Even though these slow oxidative fibers could hold up your head all day long, if you can hold your arm out to the side without moving it for 10 minutes, you're going to feel like you've had enough. So we want to compare the extremes here. And any one muscle organ can have a variety of these. And if you picture that, now you can start to see how certain individuals might even have a great predisposition for certain activities. Maybe you have a much greater composition of fast glycolytic fibers. So maybe you're going to be great at very fast bursts like sprinting. As opposed to someone else who might have a higher composition of slow oxidative fibers. They may never be able to sprint with any blinding speed, but they may be able to do a marathon in under 2 hours, whereas a sprinter may never be able to meet that sort of endurance. So it kind of opens up the thought of individual performance. And we look at any cross-section of a muscle, and we may see any sort of ratio between one and the next. Muscle undergoes a use it or lose it phenomena. If you aren't putting stress on muscle, it tends to weaken and waste. Likewise, added stress tends to see a response to that muscle organ by getting larger, stronger. It'll increase its mitochondrial content. If you're new to the gym, as I record this, it's March. People have gotten into their New Year's resolutions. Maybe they're hitting the gym for the first time in years. Some of the first responses you see when beginning a workout program are the muscles increasing in mitochondria. Some of your early strength gains are going to be because of increased coordination and increased mitochondrial content. We don't see an increase in the number of fibers. We see each fiber get a little bigger. And as we see muscles go through their movement patterns, we can actually classify their movements. First, second, and third degree levers. Now, when you're studying these, right, you only have 3 variables here. Always study the one that's in the middle. A first-class lever has the fulcrum in between the resistance and the effort. Second class has the resistance in the middle. And a third-class lever will always have the effort in the middle. All we're doing is changing out the middle component. And while we can see examples of each throughout the body, we can also use other devices that make use of these 3 classes of levers to undergo different activities. We use other terms with respect to muscle, and we'll use these even when we do our reflexes when we finish off neuro. Agonist, antagonist, and synergist, and some of these are going to be very relative terms. Picture the agonist and the antagonist as being diametrically opposite terms when it comes to muscle groups. If you are doing a bicep curl and you're flexing your bicep and moving that weight upwards, in that case, the prime mover, the agonist, is the bicep, while the antagonist is the tricep. However, if now you're doing the arm is straightening against resistance, now the tricep becomes the agonist, while the bicep becomes the antagonist. So these are relative to one another. A synergist is the muscle that aids the prime mover. At the back of your calf, you have the large gastrocnemius muscle. Well, deep to it, you also have the soleus muscle and the plantaris muscle. Much weaker, but it's assisting the same movement as the gastrocnemius. And the last thing we have to get into is not going to be lecture material, but it's going to help you with lab. You are going to be responsible for naming muscles. You may get a muscle on a diagram. You'll have to name the muscle. You'll have to describe the action. I want you to always be conscientious of the name of the muscle. Very, very, very often, the name of the muscle gives you an awful lot of information about what that muscle is, where it is, and what it does. Very rarely will you look at a name of a muscle and it give you no information. Gastrocnemius. Sure, that probably translates into something useful, but nothing you're going to be able to use in the middle of a lab exam. Orbicularis oris, though, does. Orbicularis, circular. Oris, mouth. Circular muscle that goes around the mouth. Flexor carpi digitalis. It's a flexor muscle. Digit means the fingers. It's meant to bend the wrist and flex the hands into a fist. So look at the name of the muscle, and it's going to tell you something about the muscle action, like the flexor carpi ulnaris. Specific body regions, flexor carpi ulnaris, right? Carpal bone. Muscle attachments. May tell you which bones it's hooked onto. The orientation of the muscle fibers. Terms like deltoid, meaning triangle. Trapezoid, meaning trapezoidal shape. Erectus spinous. The erectus can mean parallel. So these terms tell us a lot about the shape, size, and orientation of the fibers. How many heads? Bicep, tricep. So as you're studying the muscles, don't simply get yourself a stack of cue cards and try and do grunt memorization. Identify the muscle names that tell you all this information. And if you can read the muscle name, now you no longer need to memorize what it does. You're just simply translating. Use that to your advantage. So today we wanted to get down the microscopic anatomy of the muscle. Sure, we got into a little bit of a contractile discussion because it was difficult to talk about the structure without at least indicating the function. But our goal was to get down the anatomic arrangement, the cells, the connective tissue layers, the fascicles, myofibrils, filaments, and how the filaments orient themselves in these sarcomeres. Now we're going to go from here and look at the contractile cycle of that muscle, how we use the stimulus to excite that muscle cell, how we get the release of calcium to remove the regulatory proteins, and then how we use ATP to undergo this cyclical attaching and detaching of the actin and myosin filaments.